Hello students, I am Dr. Himanshu Arora from Department of Anthropology, University of Delhi. Today I am going to speak on the module Biotechnology and Anthropology from the paper Applied Anthropology. The learning objectives of the module is to focus on the application of biotechnology in anthropology and especially among human beings. Now, as we all know that anthropology is the study of human in all aspects, and biological anthropology, which is one of the main branch of anthropology, is the specialized discipline concerned with the biological and behavioral aspects of humans. Biotechnology is the tool of any living systems which is developed to modify products or processes for specific use by using technological application that uses biological systems. Hungarian engineer Karol Erecki first coined the term biotechnology in 1919 and later apart from agricultural and medicine, it has expanded genomics, recombinant gene techniques, applied immunology and development of pharmaceutical to therapies and food production. There are many different terms that has been developed in the field of biotechnology. Some of them are blue biotechnology. This is a term that has been used to describe the marine and aquatic applications. Green biotechnology, which is applied to agricultural processes. Red biotechnology, it is applied to medical processes. And white biotechnology, also known as industrial biotechnology. Apart from these, a new field has also been emerged as pharmacogenomics. This is the technology that analyzes how genetic makeup affects an individual's response to drugs. Such technology developed personalized medicine concept, which is very important in population variation and ethnic specificity in anthropology. Another important area is molecular anthropology, in which molecular analysis is used to determine evolutionary links between ancient and modern human populations as well as between contemporary species. Generally, comparisons are made between sequence, either DNA or protein sequence. However, early studies used comparative serology. By examining DNA sequences in different populations, scientists can determine the closeness of relationships between populations or within populations. Certain similarities in genetic makeup let molecular anthropologists determine whether or not different groups of people belong to the same haplogroup, and thus if they share a common geographical origin. This is significant because it allows anthropologists to trace patterns of migration and settlement, which gives helpful insight as to how contemporary populations have formed and progressed over time. Molecular anthropology has been extremely useful in establishing the evolutionary tree of humans and other primates, including closely related species like chimps and gorillas. While there are clearly many morphological similarities between humans and chimpanzees, such information is useful in searching for common ancestors and coming to a better understanding of how humans evolved. There are two continuous linkage groups in humans that are carried by a single sex. The first is the Y chromosome, which is passed from father to son. The other linkage group is the mitochondrial DNA. Mitochondrial DNA can only be passed to the next generation by females, but only under highly exceptional circumstances. Is mitochondrial DNA can only be passed to the next generation by females, but only under highly exceptional circumstances is mitochondrial DNA passed through males. The non-recombinant portion of the Y chromosome and the mitochondrial DNA under normal circumstances do not undergo productive recombination. Part of the Y chromosome can undergo recombination with the X chromosome, and within ape history the boundary has changed. Such recombinant changes in the non-recombinant region of Y are extremely rare. Since Crink's Neanderthal mitochondrial DNA have been sequenced, and the sequence similarity indicates an equally recent origin from a small population on the Neanderthal branch of late hominids. MCR1 gene has also been sequenced, but the results are controversial. Asian DNA sequencing has been conducted on a limited scale up to the late 1990s when the staff at the Max Planck Institute shocked the anthropology world by sequencing DNA from an estimated 40,000-year-old Neanderthal, which were derived from haplogroup H. Neanderthal branched from humans more than 300,000 years before haplogroup H reached Europe, while the mitochondrial DNA and other studies continued to support a unique recent African origin. This new study basically answered critiques from the Neanderthal side. Loci that have been used in molecular phylogenetics are cytochrome C, serum albumin, hemoglobin, mitochondrial D loop, TMRCA, which is time to a most recent common ancestor, Y chromosome, HLA DR, human leukocyte antigen D-related CD4, which is an intron, pyruvate dehydrogenase E1 alpha 1, which is X-linked, and the different X-linked loci are PDHA1, XQ213, XQ13.3, ZFX, FIX, I12RG, P1P, GK, IDS, etc. Another important advancement in the field of biotechnology is in vitro fertilization or IVF, which is a process of fertilization where an egg is combined with sperm outside the body. The fertilized egg, which is also called zygote, is cultured for 2 to 6 days in a growth medium. An embryo culture and is then transferred to the same or another woman's uterus with the intention of establishing a successful pregnancy. It may solve the problem of decaying population. IVF is a type of assisted reproductive technology used for infertility treatment and gestational surrogacy. It gives birth to the concept of controversial fertility tourism and surrogacy. Haploid loci in molecular anthropology. As I have told you earlier that there are two continuous linkage groups in humans that are carried by a single sex. The first is the Y chromosome, which is passed from father to son. The other linkage group is the mitochondrial DNA. Mitochondrial DNA can only be passed to the next generation by females, but only under highly exceptional circumstances is mitochondrial DNA passed through males. More recently, the mitochondrial DNA genome has been used to estimate branching patterns in people around the world, such as when the new world has settled and how. The researchers have discovered that as humans move from Africa's southeastern regions, the more mutations accumulated in the coding region than expected, and in passage to the new world, some groups are believed to have passed from the Asian tropics to Siberia to an ancient land region called Beringia and quickly migrated to South America. Many of the mitochondrial DNA have far more mutations and rarely mutated coding sites relative to expectations of neutral mutations. Along with many biotechnology techniques, DNA hybridization is unique. A year later, Brown and Wilson were looking at RFLP fragments and determined the human population expanded more recently than other ape populations. In 1984, the first DNA sequence from an extinct animal was done. Sibley and Elquist applied DNA hybridization technology to anthropoid phylogeny and C. Pan-human split closer than gorilla pan or gorilla human split, a highly controversial claim. The technique they used was restriction fragment length polymorphism, that is RFLP, which was more affordable at the time compared to sequencing. In 1980, W. M. Brown looking at the relative variation between human and other species, recognized there was a recent constriction in the human population, and OTA published their finds on the rapid evolution of HLA by short distance recombination. Another important concept of biotechnology is genetic engineering, which is a kind of genetic modification. It is the direct manipulation of an organism's genome using biotechnology. It is a set of technologies used to change the genetic makeup of cells, including the transfer of genes within and across species boundaries to produce improved or novel organisms. New DNA is obtained by either isolating or copying the genetic material of interest using molecular cloning methods or by artificially synthesizing the DNA. An organism that is generated through genetic engineering is considered to be the genetically modified, and the resulting entity is a genetically modified organism. Insulin producing bacteria were commercialized in 1982, and genetically modified food has been sold since 1994. Genetic engineering techniques have been applied in numerous fields, including research, agriculture, industrial biotechnology, and medicine. Enzymes used in laundry detergent and medicines such as insulin and human growth hormone are now manufactured in genetically modified cells. Gene isolation and cloning. The next step is to isolate the candidate gene. The cell containing the gene is opened and the DNA is purified. The gene is separated by using restriction enzymes to cut the DNA into fragments or polymerase chain reaction (PCR) to amplify up the gene segment. These segments can then be extracted through gel electrophoresis if the chosen gene or the donor organism's genome has been well studied. It may already be accessible from a genetic library. If the DNA sequence is known but no copies of genes are available, it can also be artificially synthesized. Once isolated, the gene is ligated into a plasmid, then inserted into a bacteria. The plasmid is replicated when the bacteria divide, ensuring unlimited copies of the genes are available. Inserting DNA into the host genome. There are a number of techniques available for inserting the gene into the host genome. This availability can be induced in other bacteria via stress, which is thermal or electric shock, which increases the cell's membrane's permeability to DNA. Uptaken DNA can either integrate with the genome or exist as a extra chromosomal DNA. DNA is generally inserted into animal cells using microinjection, where it can be injected through the cell's nuclear envelope directly into the nucleus or through the use of viral vectors. Genetic engineering has applications in medicine, research, industry, and agriculture, and can be used on a wide range of plants, animals, and microorganisms. Genetic engineering has many applications to medicine that include the manufacturing of drugs, creation of model animals that mimic human conditions, and gene therapy. One of the earliest uses of genetic engineering was to mass produce human insulin in bacteria. This application has now been applied to human growth hormones, follistim for treating infertility, human albumin, monoclonal antibodies, anti-hemophilic factors, vaccines, and many other drugs. Mouse hybridomas, cells fused together to create monoclonal antibodies, have been humanized through genetic engineering to create human monoclonal antibodies. A genetic screen or mutagenesis screen is an experimental technique used to identify and select for individuals who possesses a phenotype of interest in a mutagenized population. Hence, a genetic screen is a type of phenotype screen. Genetic screens can provide important information on gene function as well as the molecular events that underlie a biological process or pathway. While genome projects have identified an extensive inventory of genes in many different organisms, genetic screens can provide valuable insight as to how those genes function. Now let's talk about some terminologies commonly used in biotechnology. First of all, somatic cell nuclear transfer, which is also called SCNT. This is a technique in which the nucleus of a somatic body cell is transferred to the cytoplasm of an enucleated cell, which is an egg that has had its own nucleus removed. Once inside the egg, the somatic nucleus is reprogrammed by egg cytoplasmic factors to become a zygote. A primer is a short strand of RNA or DNA, generally about 18 to 22 bases, that serves as a starting point for DNA synthesis. It is required for DNA replication because the enzymes that catalyze this process, DNA polymerases, can only add new nucleotides to an existing strand of DNA. The polymerase starts replication at the third end of the primer and copies the opposite strand. In vivo DNA replication utilizes short strands of RNA called RNA primers to initiate DNA synthesis on both the leading and lagging strands. That is, DNA primers are not seen in vivo in humans. A single nucleotide polymorphism, often abbreviated to SNP, is a variation in a single nucleotide that occurs at a specific position in the genome, where each variation is present to some appreciable degree within a population. Association studies can determine whether a genetic variant is associated with a disease or trait. Tag SNPs are useful in whole genome SNP association studies, in which hundreds of thousands of SNPs across the entire genome are genotyped. Haplotype mapping. Sets of alleles or DNA sequences can be clustered so that a single SNP can identify many linked SNPs. Linkage disequilibrium. LD, a term used in population genetics, indicates non-random association of alleles at two or more loci, not necessarily on the same chromosome. It refers to the phenomenon that SNP allele or DNA sequence which are close together in the genome tend to be inherited together. A short tandem repeat STR analysis is one of the most useful methods in molecular biology, which is used to compare specific loci on DNA from two or more samples. A short tandem repeat is a microsatellite consisting of a unit of 2 to 13 nucleotides repeated hundreds of times in a row on the DNA strand. STR analysis measures the exact number of repeating units. This method differs from restriction fragment length polymorphism analysis, since STR analysis does not cut the DNA with restriction enzymes. Instead, probes are attached to the desired regions on the DNA, and a polymerase chain reaction is employed to discover the lengths of the short tandem repeats. Let's pour some light on the application of STR in forensic science and forensic anthropology. STR analysis is a tool in forensic analysis that evaluates specific STR regions found on nuclear DNA. The variable nature of the STR region that are analyzed for the forensic testing intensifies the discrimination between one DNA profile and another. Forensic science takes advantage of the population's variability in STR lengths, enabling scientists to distinguish one DNA sample from another. The system of DNA profiling used today is based on PCR and uses simple sequences or short tandem repeats. This method uses highly polymorphic regions that have short repeated sequences of DNA. The most common is 4 bases repeated, but there are other lengths in use including 3 and 5 bases. Because unrelated people most certainly have different numbers of repeat units, STRs can be used to discriminate between unrelated individuals. These STR loci are targeted with sequence-specific primers and amplified using PCR. The DNA fragments that result are then separated and detected using electrophoresis. There are two common methods of separation and detection: capillary electrophoresis and gel electrophoresis. A variable number tandem repeats or VNTR is a location in a genome where a short nucleotide sequence is organized as a tandem repeat. These can be found on many chromosomes and often show variations in length among individuals. Each variant acts as an inherited allele, allowing them to be used for personal or parental identification. Their analysis is useful in genetics and biology research, forensics, and DNA fingerprinting. There are many applications of biotechnology. Some of the applications are recombinant human insulin, almost completely replaced insulin obtained from the animal sources, like from pigs and cattle, for the treatment of insulin dependent diabetes. A variety of different recombinant insulin preparations are in widespread use. Recombinant insulin is characterized by inserting the human insulin gene into E. coli or yeast, which then produces insulin for human use. Recombinant human growth hormone HGH and somatotropin administered to patients whose pituitary glands generate insufficient quantities to support normal growth and development. Before recombinant HGH became available, HGH for therapeutic use was obtained from pituitary glands of cadaver. Recombinant blood clotting factor 8, a blood clotting protein that is administered to patients with forms of the bleeding disorder hemophilia who are unable to produce factor 8 in quantities sufficient to support normal blood coagulation. Before the development of recombinant factor 8, the protein was obtained by processing large quantities of human blood from multiple donors. Recombinant hepatitis B vaccine. Hepatitis B infection is controlled through the use of a recombinant hepatitis B vaccine, which contains a form of the hepatitis B virus surface antigen that is produced in yeast cells. The development of the recombinant subunit vaccine was an important and necessary development because hepatitis B virus, unlike other common viruses such as poliovirus, cannot be grown in vitro. Diagnosis of infection with HIV. Each of the three widely used methods of diagnosing HIV infection has been developed using the recombinant DNA. The antibody test, ELISA or a Western blot, uses a recombinant HIV protein to test for the presence of antibodies that the body has produced in response to an HIV infection. The DNA test looks for the presence of HIV genetic material using reverse transcription polymerase chain reaction. Development of the RT-PCR test was made possibly by the molecular cloning and sequence analysis of HIV genomes. Different methodologies used in biotechnology includes Southern blot. Southern blot is a method used in molecular biology for detection of a specific DNA sequence in DNA samples. Western blot. Western blot is a widely used analytical technique used in molecular biology, immunogenetics, and other molecular biology disciplines to detect specific proteins in a sample of tissue, homogenate or extract. Far Eastern blotting is a technique used for analysis of lipids separated by high performance thin layer chromatography HPTLC. The lipids are transferred from the HPTLC plate to a PVDF membrane for further analysis, for example, by enzymatic or ligand binding assays or mass spectrometry. Polyacrylamide gel electrophoresis describes a technique widely used in biochemistry, forensics, genetics, molecular biology, and biotechnology to separate biological macromolecules, usually proteins or nucleic acids, according to their electrophoretic mobility. Mobility is a function of the length, conformation, and charge of the molecule. Fast parallel proteolysis is a method to determine the thermostability of proteins that does not need previous purification and labeling. Quantitative dot blot analysis. It is a method for quantifying specific DNA, RNA, or protein molecules using a dot blot apparatus. Now, let's talk about bioinformatics. It is an interdisciplinary field which addresses biological problems using computational techniques and makes the rapid organization as well as analysis of biological data possible. The field may also be referred to as computational biology and can be defined as conceptualizing biology in terms of molecules and then applying informatics techniques to understand and organize the information associated with these molecules on a large scale. Bioinformatics plays a key role in various areas such as functional genomics, structural genomics, proteomics, and forms a key component in the biotechnology and pharmaceutical sector. Now, bioinformatics play a great role in anthropology also. With every pros there are many cons of technology. One of the disadvantages of this technology is molecular war, which is a great threat of humankind. We can also call it the black side of this technology. A number of militants are thinking about molecular war, but we should stop this to save and for good survival of mankind. Now let us summarize what we have studied till now. Anthropology is the study of holistic aspect of men, and biological anthropology deals with the biological aspects of men as a whole. Biotechnology is the concept of development and modification of individual in genomic level for further development. Now, biotechnology are using largely in anthropology, especially in biological anthropology. Different biotechnology are there: blue, green, red, white, etc. Development of molecular anthropology give a new dimension of application of biotechnology in anthropology. DNA hybridization is one of the major way to modify the target genomic area or locus. Recombinant DNA technology plays a great role in implementation of the modification for the wellness as an important methodology. Different techniques are now using for this work, that is different blot techniques, western blot, southern blot, etc. and quantitative dot blot technique. Only the great threat for us for using this very important technology is molecular war. This should be stopped for better mankind in survival. Thank you.